So now we're going to focus on the other category of saponifiable lipids, and these are the phospholipids. All phospholipids have a phosphate group in them. There are two main types of phospholipids that your book talks about: the glycerophospholipids, which have glycerol as a component, and the sphingophospholipids, which has sphingosine as a component. I'm just going to focus on the glycerophospholipids, okay? So that's what we're going to do now. Glycerophospholipids. Their main function in the human body, main function in the body, is that they are part of the membrane. They're membrane lipids, okay? Again, I mentioned this earlier, but for those of you who had a bio class, you've heard of the phospholipid bilayer of all the plasma membranes. That's what these glycerophospholipids are. Okay, so structure-wise, I'm going to focus on one specific phospholipid, which is lecithin. All right, let's focus on lecithin. Lecithin is a phospholipid that has glycerol, two fatty acids, a phosphate group or phosphoric acid group, and then it's hooked up to something called choline. So another name for lecithin is phosphatidylcholine. Oops, I can spell this right. Choline. All right. So you guys remember the structure of glycerol. With all the triglycerides, you've used glycerol. You've got two of the alcohol groups of glycerol are esterified to two fatty acids. This could be any two long chain fatty acids, any two. The only thing though, the second position has to be esterified to an unsaturated long chain fatty acid. So let's say this one is stearic, or maybe arachidic. So I have a total of 20 carbons, that's arachidic, that's fine. But this one has to be unsaturated. So it could be monounsaturated or polyunsaturated. I'm just going to choose oleic acid, which is monounsaturated, and cis. Okay, and here's CH2, 7, CH3. So this is arachidic, this is oleic. All glycerophospholipids, whether it's lecithin or any other, have two long chain fatty acids. But this second position has to be a cis unsaturated. So you're going to have a kink there. You're going to see this when we're drawing the image of a plasma membrane. That kink is shown in the diagram. All right, the third position, this third position here of glycerol is not esterified to a fatty acid, but it's esterified to a phosphoric acid. All right, so instead of having a carboxylic acid esterified to an alcohol, you now have a phosphoric acid esterified to the alcohol. So I'm going to, and normally we'll flip it over and show it on this side so that it could be a little different than this, but I don't have a lot of room here, so I'm just gonna put it over here. This is my phosphoric acid, okay? So I have an esterification between an alcohol and a phosphoric acid. HOH goes away. Phosphoric acid, by the way, is H3PO4, and it becomes this. Now, since it's an acid, a hydrogen goes away. You've got a charge. Charge makes it polar. So you're going to notice that with phospholipids, you have these very non-polar tails, and then you're going to have a polar head. Since I'm trying to draw lecithin, I need to add in choline. Choline is this amino alcohol. So we're going to have another ester bond. So Here's one ester bond, two ester bond, three ester bond. There's going to be another ester bond between this and an alcohol. And the alcohol is choline. So between this acid and this alcohol. And this over here is choline. Now, anytime a nitrogen has 4 bonds, remember from the amine chapter, it has a charge. So this is the structure of lecithin or phosphatidylcholine. I know that, what was it? I had a couple of friends who would take lecithin to help their mental abilities or help them study. And I used to tell them that taking lecithin to increase your brain power is pretty much the same as eating hair so that you could increase the hair on your head. It really, I mean, it's a substance that your body knows how to synthesize. If you have good nutrition, you can produce all the lecithin you need. But it is a very important part of every cell's plasma membrane. Anyway, back to the structure. This over here, you can see this. This is polar. This part is polar. And these two long chain fatty acids, that's nonpolar. So this is called the polar head. You can see down here, that's the polar head. And these are the non-polar tails. Okay, so what I'm going to do is I'm going to erase this and I'm going to show you how many times they will represent a phospholipid. They will, instead of drawing this whole thing out, again, you're a chemist this semester, may as well learn how to draw a phospholipid. You have 2 of the carbons of the glycerol esterified to fatty acids. The 3rd carbon of the glycerol is esterified to phosphoric acid, which is then also esterified to the choline for lecithin. In a bio class, this is what you would usually do. You would have this. Have you seen that? This is the head and the 2 fatty acid tails. What do you think this kink is? That's the unsaturated fatty acid. So what you'll see in a plasma membrane is something like this. It's a phospholipid bilayer. And remember I said phospholipids' main function in your body is they are part of membrane structure. So bilayer. So imagine that this is your cell and you've got water on the outside of the cell and water on the inside of the cell. This is this section over here. So you notice that the polar heads, the ones that have the charge, are facing towards the outside of the cell because that's polar, it's got water in it. And then these polar heads on this section of the bilayer are facing into the inside of the cell over here because this is polar also. But this interior part, this is all non-polar or hydrophobic. Why? Because fatty acids hate water and you see them like this, okay? So this is a section of the plasma membrane. And we call it a phospholipid bilayer. All right, now on your what you need to know list, I wrote, know the emulsifying properties of phospholipids. Now, as a baker, I love to bake. I'm always thinking about what's going on when I'm baking something on a molecular level. But I don't know how many of you guys bake or do anything amazing in the kitchen, but I don't do anything amazing in the kitchen, but I do love to bake. A lot of times we use an egg, and an egg, the egg yolk is filled with lecithin, and the egg yolk helps bring about the oil and water or any liquid mixture and makes a really nice emulsion. So you remember how soap emulsified the dirty oil with the water that you're rinsing your hands with? Phospholipids can do the same thing in foods. And you can see how that would potentially happen if you try this at home. If you get a beaker or just any cup and you put some water and you put some oil, the oil is going to layer right on the top. But if you crack an egg and separate out the yolk is filled with lecithin, filled with phospholipids. What's going to happen? Well, your oil droplets start to form an emulsion in the water. It won't look actually that perfect. You'll still have water on the bottom and you'll still have oil on the top, but your emulsion will start to form right over here. How does that happen? Well, the oil is nonpolar. The two tails of the phospholipid stick into the oil and the head sticks into the water. Pretty much the same emulsifying as the soap. So each oil droplet is surrounded by a bunch of phospholipids with the tails poking into the oil and allowing them to suspend in the water. So if you have a jar, put some water, put some oil, crack a couple of egg yolks, mix it really, really, really well, shake it like crazy, and then you're going to see a nice little emulsion in that interface between the oil and the water. And that's what happens in baking. Okay, our last video in this chapter deals with sterols, and then we'll just wrap this whole thing up, and I will see you in a bit.